Hello friends, welcome to my YouTube channel. Today we are discussing about Brucella species. Brucella species. Basically, Brucella species is a gram-negative coccobacilli. It should be an intracellular parasite. It should affect the animals and also infect humans. It should be cause the infection zoonotic disease of brucellosis. Brucellosis infection caused by Brucella species. It should be primarily affected by sheep, cattle, buffaloes, pigs, and other animals transmitted to human by contact with infected animals or through ingestion of their products. The human disease with various names: Mediterranean fever, Malta fever, undulant fever, remittent fever, Gibraltar fever, Cyprus fever. The disease caused by members of the genus are characterized by a number of names based on the original microbiologist who isolated the described the organisms. The various names of human disease should be isolated the microbiologist described the organisms. Next, taxonomy. Brucella belongs to family Brucellaceae. It should be a Brucellaceae family. Genus Brucella encompasses 9 recognized species, 6 terrestrial species and 3 marine species. Terrestrial species are Brucella melitensis, Brucella abortus, Brucella suis, Brucella canis, Brucella ovis, Brucella nematomiae. Marine species are Brucella depklini, Brucella pinnipidae, Brucella cecitaceae. Morphology. Morphology of the Brucella species should be a species are small gram-negative aerobic coccobacilli. It should be 0.5 to 0.7 micrometer and 0.6 to 1.5 micrometer in size. They are non-motile, non-capsulated organism, non-spore forming and non-acid fast organism. It should be Brucella's characteristics. Cultural characteristics. Brucella are strict aerobics. It should be compulsory respiration alone in aerobic respiration only. Brucella abortus is capnophilic. It should be present only in the carbon dioxide condition alone. Many strains required 5 to 10% CO2 for growth. Optimum temperature is 37 degrees Celsius, range 20 to 40 degrees Celsius, and pH should be 6.6 to 7.5. Grow on simple media through growth is slow and scanty. It should be grow only in slow and scanty. See, it should grow in simple media alone. Growth is improved by addition of serum or liver extract. Growth will be increased or improved immediately means adding of serum or liver extract. The media employed currently the serum dextrose agar, serum potato infusion agar, tripitase soy agar and tripitose agar. Culture characteristics. The addition of bacitracin, polymyxin and cycloheximide to the above media makes them selective. Erythritol has a specially stimulating effect on the growth of brucellae. On colonies are small, moist, translucent and glistering of 3 or more days of incubation. Minimum it should be solid media colonies or small and moist translucent colonies will be grow in that solid media. Glistering after 3 or more days of incubation. Glistering should be visible in 3 or more days of incubation. The liquid media growth in uniform. Solid media should be 3 to more days incubation. On liquid media should growth uniformly. Cultural characteristics of culture media. Biochemical reactions. No carbohydrates are fermented. Catalase and oxidase should be positive. Neotomy and Brucella ovis which are negative. Nitrate reduction positive. Imbic all negative. Indole methylate Vargas prostate citrate utilization should be negative. Urease test should be positive. Classification. Brucella may be classified into different species based on Based on the classification, lysis by specific bacteriophage, CO2 requirements, H2S production, hydrogen production, oxygen production, carbon dioxide production, agglutination by monospecific sera, sensitivity to dyes, biotypes, Brucella abortus 7 biotypes, Brucella melitensis 3 biotypes, Brucella suis 5 biotypes. Portal of entry. Portals of entry. The Brucella infection should be affected humans or the animals in particular portal of entry. Entry of human pathogen should be oral entry, aerosols, percutaneous infection. Oral entry. Ingestion of contaminated animal products, often raw milk or its derivatives, contact with contaminated fingers. It should be entry point of human infected animal to humans. Aerosols, inhalation of bacteria, contamination of the conjunctiva. Percutaneous infection through skin abrasions or by accidental inoculation. Brucella infection. Brucella structure should be gram-negative bacilli. Pathogenesis. Human infection may be of three types. Latent infection, pathogenesis. Human infection. Brucella infection should be three stages. These are latent infection, acute or subacute brucellosis, and chronic brucellosis. Acute brucellosis. Acute brucellosis is mostly due to Brucella melitensis should be caused the infection of acute brucellosis. It is usually known as undulant fever, but this is misleading as only some cases show the undulant pattern. It is associated with prolonged bacteremia and irregular fever. It should cause the bacteremia infection. Presence of bacteria in blood is called bacteremia and irregular fever. Symptomology is varied, consisting of muscular and articular pains, asthmatic attacks, heart attack. It should be a heart attack infection and articular pains, joint pains, nocturnal drenching sweats, exhaustion, anoxia, constipation, nervous irritability, and chills. The usual complications are articular, osseous, visceral, and neurological. Subacute brucellosis. It may follow acute brucellosis. Blood culture is less frequently positive. Chronic brucellosis. Chronic brucellosis, which may be non-bacteremic, is a low-grade infection with periodic exacerbations. The symptoms are generally related to the state of hypersensitivity in the patient. Symptoms should be hypersensitivity in the patient. Common clinical manifestations are sweating, lassitude, and joint pains, minimal or no pyrexia. The illness lasts for years. Clinical manifestations: fever, night sweats, malaise, anorexia, arthalgia, fatigue, weight loss, depression. That's all should be clinical manifestations of symptoms of Brucella species. Epidemiology: Brucellosis is a zoonotic disease. Brucellosis is worldwide distribution and endemic certain areas such as Mediterranean countries. It should be a zoonotic disease. It should be spread worldwide. Human infection: direct or indirect contact with infected animal tissue. Yeah, direct contact or indirect contact with infected animal tissue. It should be spread via humans. Person to person transmission, rare in circumstance, implicating sexual contact, tissue transfer including blood and bone marrow. Laboratory acquired brucellosis, accidental ingestion, inhalation injection, mucosal and skin contaminations. Laboratory diagnosis: specimen blood, urine, sputum, breast milk, lymph node, biopsy, and bone marrow aspirate. Laboratory methods of diagnosis include culture, serology, hypersensitivity test, molecular testing. These are all the laboratory diagnosis of Brucella species. Serology. Most serological studies for diagnosis of Brucella is based on the antibody detection. This includes serum agglutination test SAT, standard tube agglutination, Rose Bengal test, slide agglutination, ELISA, complement fixation test, indirect counts, immune capture agglutination, whole cell preparation of Brucella antigen are used in IFA agglutination. Purified LPS protein extracts are used for the ELISA. Laboratory diagnosis in animals. This includes rapid plate agglutination test and Rose Bengal card test. Milk ring test. This is a frequently used serological test for the demonstration of antibodies in the milk of an animal. Contaminated milk or contaminated animals' properties should be affected humans. Uh, should be affected the humans brucellosis, brucella infection. It should be avoided means the milk ring test should be compulsory doing for that avoid of brucellosis. This is a screening test used to detect the presence of Brucella infection in infected cattle. In this test, concentrated suspension of killed bee abortus or B. militancies stained with hematoxylin is used as an antigen. The test is performed by adding a drop of colored Brucella antigen to the sample of whole milk in a test tube. Take a single test tube and add a colored Brucella antigen sample, and then it should be mixed suspension incubated water bath 70 degrees Celsius to 70 degrees Celsius for 45 to 50 minutes. Colored Brucella antigen should be added after it should be mixed well, then place the water bath 70 degrees Celsius for 45 to 50 minutes. In the positive test of antibody present in the milk, the bacilli are agglutinated and raised with cream to form a blue ring at the top, leaving the milks unstained. Leaving the milk unstained. The positive test of milk ring test after adding the antibodies, the present the milk. If antibodies are present in the milk, the bacilli are agglutinated and raised a cream to form a blue ring at the top. The milk will be white and the Brucella bacilli should be a ring, blue color ring at the top of the test tube. It should be a milk ring test result. The negative test, the milk remains uniformly blue without formation any colored ring. Treatment. Brucella infection should be affected worldwide. It should be a zoonotic disease. How should we control for the infection of brucellosis? Brucella are sensitive to a number of oral antibiotics and aminoglycosides. The combination of tetracycline and doxycycline is effective against most species of Brucella. Thank you.